So here's a reminder of the type of nephrons we're talking about when we talk about the nephron loop and the medullary gradient that's important for this osmotic flow. It's these juxtamedullary nephron that have a vasa recta capillary system. The vasa recta is what surrounds these long nephrons that are in the renal medulla. So we are right down near the renal pelvis. So this is what we're talking about with the 1200 milliosmoles down here. These cortical nephrons that are surrounded by paratubular capillaries don't have that same function. A bit, actually, to the nephron loops do the same thing, but they are not able to the same extent. They're not reaching that 1200 milliosmoles to allow for as much water reabsorption and solute reabsorption. So they're able to do less, reabsorb less. So this is the entire process. Again, this would be a juxtaglomerular nephron. And we know that because we're all the way down to the inner medulla. These are the only nephrons that reach that far down into the inner medulla near the renal pelvis. So we've got the filtrate entering this nephron loop. That filtrate is 300 milliosmoles, so isoosmotic to the blood and to the ISF at this location. As we start to drop down through the outer medulla, inner medulla, deeper and deeper into the medulla, the osmolarity in the ISF starts to go up. ISF osmolarity increasing as we go down physically. That's going to result in water reabsorption down that osmotic drive. So at this point, the filtrate entering is like 500. As it enters the renal medulla that is 600, water flows out down its osmotic gradient due to the osmotic gradient and concentration in the medulla itself. So this is where water is reabsorbed, first concentrating the filtrate, and then we're going to go back up and dilute it. So here is the maximum concentration in terms of osmolarity that our filtrate's going to get. This is going to depend on how far that loop dips into the medulla. It's going to be equal to the osmolarity in the renal medulla wherever this nephron loop dips down to. So again, this would be a vasa recta that goes all the way down into near the renal pelvis. A cortical loop would not reach that same max concentration. As we ascend up the ascending loop, we're going to have the reabsorption of salts, and that's going to occur both through passive transport, some here, down the electrochemical gradient of those solutes as we go up and we have decreasing medullary osmotic concentration. There is a drive for those solutes out. There's also a drive for water in, but that's not going to happen. We don't have aquaporins there. We don't want there to be because we want to reabsorb stuff. That's what the nephron loop is for. Otherwise, we'll get dehydrated. So reabsorption is passive and then actually occurs via active pumping. Active is pumping, right? That's a redundant thing to say, of salts. So the sodium, chloride, potassium, that's how we get a osmotic pressure, an osmolarity in this filtrate that's lower than the ISF, right? Right here is just 400. How can it be lower in here than it is out there? Only through active pumping, right? Otherwise it would be in equilibrium. We're pumping out more solutes using ATP to lower the filtrate concentration to even lower than it would be if we didn't have active pumping. That's what allows this to be lower osmolarity on the ascending loop than it was in the descending, and ultimately reach 100, which is very low osmolarity. From there, our urine can be formed anywhere between 100 and 1200 milliosmoles. That regulation we'll talk about later. Entering the distal collecting duct, we're at 100 milliosmoles because of that nephron loop. First concentrating the filtrate, then diluting it. Through that process, we reabsorb water, then salts. Pretty darn cool system, if you ask me. I know you're wondering what proteins are doing this. Well, one is going to be aquaporins. I mentioned there's aquaporins present on this descending loop, but not the ascending. Aquaporins facilitate water movement, right? So their water can move more quickly across the plasma membrane if there are aquaporins present. So that's all we need there. What about this active transport over here? Let's zoom into the ascending limb. Here we are, ascending limb, and this is the thick portion where the most active transport occurs. Okay, active transport, what do you think? One thing we're gonna need is a sodium-potassium pump. That's going to allow 2 potassium in, 3 sodium out. Now, we've got some other fun proteins too. One is called let me draw it first, sodium, potassium, chloride, 2-chloride, co-transporter. It's going to do exactly what it sounds like, which is move sodium in, potassium in, and 2 chlorides in. This is actually a pump that does this, a co-transport pump. And once these are in here, we can have diffusion. Do we have diffusion? Well, diffusion across the cytoplasmic space. And then we've got another protein that allows potassium and chloride out, another co-transporter. This co-transporter, SymPort, same direction, is passive transport, so it's not a pump. Does not require ATP, and you know that because we're going down our concentration gradients, right? Chloride moving out, potassium moving out, that's down our concentration gradients.